So after the blotting techniques, I'm going to go into enzyme-linked immunoabsorbent assays or ELISAs because I would say it's a kind of similar um, theme as what we just learned. So with what we just learned about specifically Western blotting in relation to proteins. So enzyme-linked immunoabsorbent assays are usually used to determine if a protein binds to its substrate or to also determine the concentration of the substrate. And so usually for determining the concentration, that's usually determining from antigens and antibodies that they usually use antigens and antibodies for ELISAs in these different samples. And there are different types of ELISAs. So one is the direct ELISA, in which first the antigen is fixed on the surface or the well plate. So actually have at the bottom here are some diagrams for the 3 different types of ELISAs that we're going to discuss right now. And also this is generally what you will see like ELISAs on. So this is the well plate, and then as you can see there are some darker shades and lighter shades that usually indicate how concentrated the substrate is. And so with first the direct ELISA, the antigen is fixed on that well plate, and then liquid with primary antibody is then washed over the plate, and then the reporter that is bound to that primary antibody that usually binds to that antigen that you have on the plate produces that color change. And so we see that here. And so that's direct ELISA. Indirect ELISA is when first an antigen is fixed on the well plate like the direct one, except that we have an additional step. So after we add the primary antibody over the plate, we also then have a secondary antibody that's specific to the primary antibody that's also washed over the plate. And then it ends up being the secondary antibody that has the reporter that then produces a color change. So indirect just has that extra secondary antibody step. And then the third ELISA, the sandwich ELISA, is first when the antibody is fixed. So this is a little different because the antibody is fixed on the well plate instead of an antigen like the first two. And then liquid with the antigen is washed over the plate. So hopefully the antigen is binding to the antibody. And then we have an additional detecting antibody that binds to the antigen that we just put there. And then we also have another secondary antibody specific to that detecting antibody that has a reporter bound to it that produces another color change. So that's the slight difference between each of them. So direct, it's just the primary antibody. Indirect, it's based on that secondary antibody. And sandwich ELISA, because you have these two antibodies sandwiching the antigen and then also having that extra secondary antibody producing the color change. So now we're going to go into another question. And so we have here 100% saying B. That's great because the answer is B. And so just to recap quickly then, we're asking in this question which methods could be used to measure RNA expression. And so we know that specifically northern blottings are specifically for RNA expression. The reason why the other answer choices are not quite right is because southern blotting is for DNA sequences or DNA expression, and then Western blotting and ELISAs are both for proteins as opposed to just RNA.